and share the screen. So today is October 14th, and we're going to be working on chapter 4. There is no lab today, but I will be logging in from, what the heck is it, 6:30 to 6:45 to answer questions. Please read the unknown project instructions. You can find that file in Canvas, and it's either in week 4 or week 5. We will talk about the unknown project next Tuesday in the lab. All right, any questions about any of that? If not, let's begin. I've got this scheduled for a week and a half. We probably won't need a week and a half, in which case we'll start chapter 5 a little early. All right. Oh, come on. So I had that we had talked about the atypical cell walls. Is that correct? And then the families Mycoplasma and Mycobacterium have atypical cell walls in the domain bacteria. And the Archaea are a different domain, and they have different cell walls as well. Realize the Mycoplasmas, they don't even have a cell wall. So you can damage the prokaryotic cells, and if you damage their cell walls, they'll be susceptible to osmotic lysis. You can damage their cell walls with lysozyme. It's actually an enzyme you can find in your skin, as well as some of your mucous membranes, like your eye, the tears of your eye. It's an enzyme which digests the disaccharide and peptidoglycan. Then you can use this enzyme to remove the cell wall of bacteria. They even have a term for a wall-less gram-positive cell and a wall-less gram-negative cell. You don't need to know these terms. I'm not going to quiz you on that. And I'm not even sure why they're a different name, but they are. And then you can also damage the cell wall by using penicillin and the penicillin-like antibiotics. Remember, penicillin inhibits the peptidoglycan bridges in peptidoglycan, so that when the cell wall is there, the peptidoglycan is laid down, but it's not connected together. And so that makes a hole in the cell wall, and the cytoplasm, I guess the cell membrane can protrude out of that hole. Any question about any of that? Does anybody know why we have lysozyme in our skin, or on our skin, whatever you want to call it, and in the tears of our eyes? Anyone know? Anyone have a guess? Why would we have an enzyme that digests the cell wall of bacteria?
Does it have something to do with salt?
Yeah, it has something to do with cells, meaning the enzyme would digest the cell wall of bacteria, and then the cell would be susceptible to lysis. And that's actually how penicillin works. So why do we have that enzyme? Come on, you guys. It's actually right down here. It's a enzyme the body makes to defend itself against bacteria. And if you think about it, you wouldn't want bacteria in your eyes. And so we secrete that into our tears. For damaging the prokaryotic cell wall, and I guess we're really only talking about bacteria, the gram-negative bacteria are not as susceptible to gram-positive bacteria to most agents that would damage the cell wall, such as penicillin G. What happens is penicillin G cannot get through the outer membrane of the cell wall. And that's true for some other antibiotics, like cephalosporin and the cephalosporin family. It doesn't work on gram-negative bacteria. But some detergents can't get through the gram-negative outer membrane as well. And so the detergent isn't very effective on gram-negative cells. Now, admittedly, these detergents aren't real strong detergents, so they're not that great on gram-positives. Now, when I say that gram-positives are more susceptible than gram-negatives to penicillin G and a few other antibiotics, that's not to say that the gram-negatives are not susceptible to antibiotics. We can apply other antibiotics like streptomycin, which gets right through the outer membrane of the gram-negative cell. And then furthermore, ampicillin, which was, I think, the third antibiotic in the penicillin family that was discovered and certainly made, ampicillin itself works on both gram-negatives and gram-positives. So you can take ampicillin against a gram-negative infection, but you cannot take penicillin G against a gram-negative infection. Any question about any of that? So when you come across an answer or a question saying, are gram-negatives less susceptible than gram-positives? It is, yes, but just a little bit. And then don't tell me the gram-negatives are not susceptible, because they are susceptible to a large range of antibiotics, as well as other antibacterial agents. Like, what's the thing they used to use? Iodine. They used to use that on surgical patients all the time. Gram-negatives are susceptible to iodine. Of course, iodine is hardly used at all in the hospital anymore because they have other antibacterial agents that they use, which I think are better than iodine. All right, any question about any of that? All right, if there's no questions, let's move on to talking about the plasma membrane. You should know that all plasma membranes of the bacteria and the eukaryotes, as well as the archaeobacteria, are a bilayer. Now, for the eukaryotes and the bacteria, it is a phospholipid bilayer, where one phospholipid is arranged this way with the head on the outside of the membrane, and then a head on the inside of the membrane, and then the two fatty acid tails are arranged on the inside of the bilayer of the membrane. They want to be on the inside because they don't want to be around water, and outside the cell, it's mostly water. If you think about a red blood cell floating in the blood is mostly water, as you are as well. And so you want to have the head on the outside and then the head on the inside because the cytoplasm inside the cell is mostly water. Now, the phospholipid is the main molecule in the cell membrane as well as all other membranes, but it's not the only molecule. There are also proteins which are integral to the functioning of the membrane. And there are three types of proteins. There's peripheral proteins, integral proteins, and transmembrane proteins. A peripheral protein is a protein which is found on one side of the membrane or the other. Any question about that? So that's why it's peripheral. It's found on the periphery of the membrane. An integral protein and a transmembrane protein spans the membrane, like a channel protein or a transport protein. All right, any question about this portion of the plasma membrane? You should know that the plasma membrane is a fluid. And it always amazes me when I look at my hand, it's held together by cells, which are stuck together by the cell membrane. And that membrane is a fluid. Any question about that? How fluid it is? It's about as fluid as olive oil. And if you don't know, if you put olive oil in the refrigerator, it will largely become solid. But at room temperature, olive oil is a fluid. It's sort of viscous. It's thicker than water. And we have a fluid mosaic model, a model to describe how the plasma membrane works. The model states that the cell membrane, as well as every membrane, is a mosaic of phospholipids and proteins. It's as fluid as olive oil, and the proteins can move around to function. Many of the proteins are freely able to move in the membrane. And those of you who took A and P, you'll remember that when they were talking about, oh gosh, what the heck is that? The signal proteins, when a ligand binds to a protein, the proteins tend to move together, come together in the cell membrane. And then that region of the cell membrane will be brought into the cell, bringing in the signal which bound to the receptors. Any question about that? Now, not all proteins move. Is that in my next slide? No. There are some proteins which do not move. These proteins are usually bound to either the cytoskeleton inside the cell or to an extracellular substance like the bone or the marrow or when was in collagen. They can be attached to an extracellular substance outside the cell. And if it's attached to a like an the intracellular skeleton, the cytoskeleton, then the protein doesn't move. It's anchored in place by the cytoskeleton. Any question about any of that? So the proteins can move, but if they're anchored to something, either external to the cell or internal, in which case it's internal, it'll be to the cytoskeleton. Is that always true? As far as I know, it's always true. in which case the protein won't move. It's not free to move around. The phospholipids themselves can move and rotate laterally. First of all, they can, let me blow that up a little. The phospholipids can rotate this way, but they can also move around. Oops, didn't mean to do that. Oh, I see what happened. I lost the blow up. So they can move around this way, and the water actually takes advantage of that to get inside the cell. Water can't move through the fatty acid tail portion of the cell membrane. And so water molecule will come up to the cell membrane and just wait for a phospholipid to move over. And then the water takes that place. And then it can't get through this portion because of the nonpolar fatty acid tails of the phospholipid. But the water molecule just waits for this phospholipid to move, and then the water can move inside the cell. Let's see, any other points about that slide? No. All right, any questions? The plasma membrane shows selective permeability, and that means it allows the passage of some molecules, but does not allow the passage of other molecules. No large molecule can move through the plasma membrane without the help of a transport protein. And that would include a protein trying to come into the cell, as well as an amino acid, and even molecules as small as glucose or fructose cannot get through the plasma membrane. Ions either do not move across the plasma membrane, or if they move very, very slowly. And that's because ions are positively charged and they do not like moving through the nonpolar region of the lipid bilayer of the phospholipid membrane. Lipid molecules can move through the cell membrane or any other membrane. However, if you think about it, a cell right here, it's mostly going to be surrounded by water and molecules that can dissolve in water. So a cell in the blood is not going to see a whole lot of lipids. There are some lipids in the blood, but there's not a whole lot. Most of the molecules are water soluble. But the point is, a lipid can move through the cell membrane, and that's because they have no trouble with the phosphate portion, and then they can move right through the nonpolar fatty acid tail. One of the main lipids in your blood is cholesterol, but there are also, high density lipoproteins, and that's not really a lipid, that's a lipoprotein. So it's a protein attached to a lipid. Any questions about any of that? Smaller molecules can move through the cell membrane. The main molecules you need to know about that can move right through the cell membrane are oxygen and carbon dioxide. And they just diffuse right through the cell membrane. Of course, oxygen diffuses into the cell, carbon dioxide diffuses out. Any questions? Is there anyone still here?
Yes.
I'm alive.
Okay.
You
I
have
was just
8 of us.
wondering. Plasma membrane also has a number of specific I don't know, structures or enzymes in it, especially for the bacteria. For the bacteria, as well as the archaebacteria, that's where we find the enzymes for making ATP production. We do not see this in eukaryote membranes, but in bacteria and archaebacteria, they contain the enzymes for making ATP. And we'll talk a little bit more about that in a later lesson. For the photosynthetic bacteria and the photosynthetic archaebacteria, that is where we have the photosynthetic pigments found in these photosynthetic cells. Now, this is for the photosynthetic bacteria and the photosynthetic archaea, not the photosynthetic eukaryotes. Plants and protists, which are photosynthetic, do not have the pigments for photosynthesis found in the plasma membrane. If the bacteria is photosynthetic, it will have the photosynthetic pigments, come on, in the plasma membrane in pockets of the plasma membrane. And you can also call them foldings. They even have a name for that, chromatospheres or thylakoids, after structures that we see in the plant, meaning it's not in the plant, it's in bacteria, and it's not the thylakoid disc, it's the thylakoids. But there you can see the chromatospheres, foldings of the cell membrane. What happens is the cell membrane goes like this, and then, maybe I can draw that better than anything else. So instead of being a cell membrane like that, the cell membrane goes like this. Let's see if I can do this well. And then it folds. Yeah, that's pretty good. You get the idea? And there's the cell membrane. So that's what makes the chromatosphere. Any questions about that? Let's see, where's my, oops, lost it. So that's what thylakoid is. And that's where the photosynthetic pigments are. If you damage the plasma membrane, that will have the cell contents leak out of the cell and will kill the cell. You can damage the plasma membrane with alcohols, quaternary ammonium compounds, and one antibiotic, the polymyxin family of antibiotics. And I think, let's see, we're not talking about antibiotics in this class. So it's polymyxin B is the specific antibiotic they give to patients to damage their plasma membranes. The movement of materials happens across the membranes. The movement of material across the plasma membrane is critical for cells. There are two processes that cells use to get things to move across the membrane. There's a passive process, and if the cell uses a passive process, it doesn't cost the cell any energy. And then there's an active process, and if the cell uses an active process, it's literally pumping the molecule it wants across its membrane, and that costs the cell energy. The energy is required for an active process because the molecule is moved against its concentration gradient, meaning if it's moved into the cell, then there would be more molecules in the cell than are outside the cell. And the cell is pumping the molecule from an area of low concentration, moving it to an area of high concentration. And that requires energy to do that. It also requires a protein to pump it. We'll talk about that in a little bit. And the energy the cell gets to move that molecule comes from ATP. The transport protein requires the energy from ATP to move or pump the molecule across the membrane. And remember, we're talking about an active process into the cell, but it could be an active process outside of the cell. Trying to think of when that would ever happen. The only thing I can maybe think of is that you would take a drug and the cell doesn't want that drug, and there's more drug in the blood, and it does move into the cell. The cell doesn't want it, so it could pump that drug outside into the blood, and that would be against the concentration gradient. The pumping of material outside the cell as an active process is actually fairly rare. Any questions about any of that? All right. For the passive processes, the molecule is moving down its concentration gradient. Like oxygen will be more in the blood, higher in the blood than in the cell. And so the oxygen will move into the cell by a passive process, in a simple diffusion actually. And it's moving from areas of high concentration to areas of low concentration. Any question about that? A passive process can happen by simple diffusion, which I just described, but it can happen by facilitated diffusion, where a solute moves into the cell through a protein channel, but it's moving from areas of high concentration to areas of low concentration. And then the third passive process is osmosis. And osmosis is water moving across the cell membrane. Water is a solvent, not a solute. And the water will move into the cell by osmosis if the water is higher outside the cell than inside the cell. Okay, so osmosis is really either simple diffusion or facilitated diffusion, but it's a solvent, not a solute. Any question about any of that? All right, let's talk about the passive processes a little bit more detail. Remember, if it's a passive process, the molecule moves down a concentration gradient, moving from an area of high concentration, where you put the dye, to an area of low concentration, where there's very little dye and mostly water. Simple diffusion happens in our cells mainly with oxygen and carbon dioxide. It can happen with other molecules as well, but these are the two we need to worry about most, because oxygen will diffuse out into the cell and carbon dioxide will diffuse out of the cell. Any question about any of that? Facilitated diffusion happens when a molecule moves down a concentration gradient. So it's higher outside the cell and then moves into the cell where the concentration gradient is less. If it's moving through a channel protein, that is facilitated diffusion. It's facilitated because we have a protein facilitating the movement of that molecule. And I will call it a channel protein. This slide also calls it a transporter protein. Don't get it confused with a transport protein because that term is used with active transport. And that is not facilitated diffusion. Active transport is where the molecule moves from areas of low concentration to areas of high concentration. Facilitated diffusion, the molecule moves from areas of high concentration to areas of low concentration. Now this slide is showing you with glucose, and I'm going to tell you don't memorize that glucose does it this way, because glucose is a molecule which uses a protein which switches its behavior, so it can be acting as a facilitated diffusion protein, but then it can also switch its behavior and act as an active transport protein. So glucose isn't a good one to remember. What you could remember is instead of glucose is fructose moves into the cell only by facilitated diffusion. Fructose can't move in by diffusion because fructose can't move through the lipid bilayer of the plasma membrane, specifically the nonpolar fatty acid tail portion of the bilayer. So fructose can only get into the cell by a facilitated diffusion through a facilitated protein that I'm going to call a channel protein. And fructose, when you eat fruit, you'll get it fructose at high concentration in your gut, and it'll be lower inside your cells. And then fructose will move into the cell by facilitated diffusion. What does that mean? It means that you will never digest all of the fructose, because once the fructose becomes equal outside the cell as inside the cell, no more fructose will move into the cell. And I'm talking about the net movement of fructose. There will be no net movement of fructose into the cell. So when you eat fruit, you will never digest all of the fructose. Any question about any of that? When you eat glucose, or let's say a candy bar, which contains sucrose, which can be broken down into glucose and fructose, will the glucose be entirely dissolved, or will some of it stay in the gut? Okay, that's a yes or no question, people. Somebody's going to have to guess.
No.
No is correct. That is not all of the glucose that is, let me see about that now. All of the glucose that you eat will be digested and brought into your cells. The reason why that's the case is that glucose has an active transport protein. And even if there's only one molecule of glucose outside the cell, that active transport protein will move that glucose into the cell, even though there's higher concentration of glucose inside the cell. So when you eat a candy bar or some other sweet, you will digest all of that sugar. But when you eat a piece of fruit, you will never digest all of the fructose. And that's just the way our cells are. Any question about any of that? All right. In both simple diffusion and facilitated diffusion, the solutes are transported, and the transport of the solute happens down the concentration gradient, meaning the molecule moves from areas of high concentration to areas of low concentration. Osmosis also moves from areas of high concentration to areas of low concentration, but osmosis is the net movement of water across a selectively permeable membrane. So in order for osmosis to happen, it has to be the movement of water across a membrane. If the water just moves from one area of soil to another area of soil, and there's no membrane in between them, that's not osmosis. That would be the diffusion of water. But when water moves across a membrane, we call it osmosis. Remember, water is a solvent, not a solute. Now, when we're talking about the movement of water, you have to realize that the concentration of the water will be the inverse of the concentration of the solute, meaning when the water is high in concentration, the solute will be low in concentration. In reverse, when the water is low in concentration, the solute will be high in concentration. Another thing to remember is that we talk about the tonicity when we're talking about the movement of water across the membrane, but the tonicity is defined by the concentration of the solute, not by the concentration of the solvent. So even if the water is the thing moving across the membrane, we define it as isotonic or hypotonic or hypertonic by the concentration of the solute. Any question about that? Make sure I talked about all of that. Yes. All right. So a cell, especially a bacterial cell, can experience three kinds of osmotic solutions. There's isotonic, hypotonic, and hypertonic. Now you should realize that each of these terms are relative. So it's correct to state that the solution outside the cell was hypertonic relative to the solution inside the cell. Meaning if we just had water in some soil in a container, in one area the soil was dry, the other area the soil was wet. And so what would that be? You can't say that the wet soil is hypotonic to the dry soil. I suppose you could, but we don't, because we use these terms when we have a membrane or a, what do you call that? A man-made membrane, like a, I don't know, plastic bag with holes in it or something like that, with pores in it. What is that called? Plastic-like structure with pores in it so that water can move across it.
Are you talking about like a sponge?
Yeah, no, that would work too, but something like a tubing. Okay, so if we had tubing and then we stick it inside of a beaker, the fluid inside the tubing could be isotonic to the fluid outside the tubing. That's what I'm trying to say. Usually we'll talk about it with the cell, in which case it's the cell membrane. All right, so if the cell is in an isotonic solution, the concentration of the solute outside the cell is equal to the concentration of the solute inside the cell. So that means there's no net movement of water into the cell. While we're saying no net movement of water, it doesn't mean that a water molecule never moves from outside the cell to inside the cell. But if one does move inside the cell, another water molecule from inside the cell moves outside. So there's no net movement of water when the cell is in an isotonic solution. And as you notice, like I said, the tonicity is defined by the concentration of the solute, not by the concentration of the solvent. But usually it's the water that does the moving across the plasma membrane. In a hypotonic solution, the concentration of the solute outside the cell is lower than inside the cell. That means there's more water outside the cell, and so water will move into the cell. That will swell the cell membrane. And if the cell does not have a cell wall to protect it, the cell membrane will expand until it will eventually burst. And then the cytoplasm will leak out of the cell, and that will kill the cell. Now, our blood cells do not have a cell wall. So if you were to put them in water, water would move into the cell, and then the red blood cell would lyse. However, most bacteria do have a cell wall, so that most bacteria, if you were to put them in water, water would only move into the cell up until the point where the cell membrane moves up against the cell wall. Then the cell membrane can't move any further, and that means no more water can move into the cell. Any question about that? And this is actually how penicillin G works, is that it disrupts the peptidoglycan links in the cell wall. And that way water can move into the cell, and then the cell membrane can protrude out of the hole that penicillin G made, and then that will eventually lyse the bacteria. That term is called osmotic lysis. Most bacteria live in a hypotonic solution where there's more water outside the cell than inside the cell. And I shouldn't say all bacteria, but many bacteria. Like, for example, most bacteria that invade your blood and then cause a blood infection, there will be more water outside the cell than inside the cell. And that would be a hypotonic solution. And that's why penicillin G would work in that case. All right, any question about any of that? There's also a hypertonic solution where the solute concentration outside the cell is higher than the solute concentration inside the cell. That means that water will leave the cell because there's more water inside the cell than outside the cell. This will cause shrinkage of the cytoplasm. The cytoplasm will pull away from the cell wall, and it may lead to plasmolysis, where the cell will die from losing too much water. Any question about any of that? This is what happens when you open up a bottle of molasses, and then the bacteria falls into the molasses from the air, and then you take the molasses and you put it at room temperature. Why the molasses does not spoil is the bacteria, when they fall into the molasses, water will leave the bacteria, and that will cause plasmolysis, the shrinkage of the cytoplasm away from the cell wall. So much water will leave the bacteria, then the bacteria will generally die. Now, an endospore can survive, but most common bacteria like E. coli would die. Any question about any of that? All right. So the passive processes are okay when the concentration of the substance required by the cell is higher outside the cell than inside the cell. But surely that often doesn't happen. If you think about it, the cell is going to want glucose. And glucose generally is not going to be higher outside the cell than inside the cell, unless the cell happens to be around, candy or something, like you eat candy in your gut and something like that. But bacteria in the ocean, for example, how often is it going to see glucose outside the cell? Or bacteria in the desert, how often are they going to see glucose? Not very often. And when they do see glucose, it'll be higher inside the cell than outside the cell. So passive processes aren't going to work. We have to move the material against its concentration gradient, and that is going to require active transport. So we have active transport to move a molecule against its concentration gradient. That's going to require energy. The cell is performing work, moving the molecule against its concentration gradient. Cells use active transport to pump the solid molecules against the concentration gradient. And we have, in order to do that, to do active transport requires, one, a transport protein to move the molecule, to literally pump it across the membrane, generally the cell membrane. Transport proteins typically are molecule specific, meaning if we have a transport protein that can transport glucose across the cell membrane, that is all that one transport protein can do, all it can move. To move, let's say, galactose across the cell membrane, you need another protein. Now, lactose is a transport protein. Any question about any of that? All right, the active transport is going to require energy to move the solute molecule against its concentration gradient. And that energy is going to come from ATP, adenosine triphosphate. Any questions about any of that? Any questions about active transport? In bacteria and archaea, we have a special active transport. So it is active transport, but it differs because the molecule that is moved is chemically altered when it is transported. So let's say, for example, glucose is transported outside the cell, inside the cell. If it happens by active transport, then the glucose is not changed. But for bacteria and archaea, we can move glucose into the cell by group translocation. Group translocation happens by a specific transport protein. There has to be a high energy phosphate compound, such as PEP, and that's got a phosphate on the end of it. And then you have to have your molecule, which is transported. And when that molecule is transported, the molecule doing the transport chemically alters that molecule, and that helps that molecule move into the cell and allows the cell to easily accumulate that substance. For example, when glucose is moved into the cell by group translocation, the glucose gets a phosphate molecule transferred onto glucose. And so instead of when it's moved into the cell, instead of moving into the cell as glucose, it'll move into the cell as glucose 6-phosphate. Glucose is chemically altered in the process of group translocation. Why do bacteria do this? Well, think about it. Let's say there were 10 bacteria or 10 glucose, I guess we should say 11, but we'll say 10 just to make it easier. Glucose molecules outside the cell. And there's one glucose molecule inside the cell. Is that right? No, that's not right. Let me start all over. Let's say there's 10 glucose molecules outside the cell and 100 glucose molecules inside the cell. Okay? So to move it into the cell, which the cell will want, because the cell will want the glucose, it has to move it against a concentration gradient. If it moves the glucose into the cell by active transport, you're moving one molecule, which is at a concentration of 10, to a concentration of 100. And that's difficult to do because the concentration gradient is so big, so high. But if we move it into the cell and make it glucose 6-phosphate, that would be easier to do because there's glucose outside the cell at 1 in something like 10 concentrations and 100 inside the cell. And we move it now into the cell and transfer it and make it glucose 6-phosphate. We now have 100 glucose molecules inside the cell and 1 glucose 6-phosphate. You see why that is easier for the cell to move it into the cell. Typically what the cell will do is, if it's trying to move a molecule in, and let me see, let's say there are 2 molecules of glucose outside the cell, and we'll say 12 molecules of glucose inside the cell. The cell will want to move it, let's say 12 molecules of glucose inside the cell and 12 molecules of glucose 6-phosphate inside the cell. And there's 2 outside. So we'll move 1 in with active transport. Now there's 13 molecules of glucose in the cell and 12 molecules of glucose 6-phosphate and only 1 molecule of glucose outside the cell. The next time the cell moves it in, it'll move it in as group translocation. And now we have 13 molecules of glucose 6-phosphate inside the cell and 13 molecules of glucose inside the cell. And you can see that because the cell has 2 ways of moving it in, it can more easily accumulate substances inside the cell, because it would be easier moving it from an area of 1 to an area of 12 than moving it from an area of 1 to an area of 13. Does everybody understand that? All right. So it does allow the cell to accumulate substances a little more easily. Group translocation, let me reiterate, is only found in the prokaryotic cells. The eukaryotes do not have group translocation. Any question about any of that? All right. If not, let's move on to cytoplasm. Cytoplasm is the substance inside the cell membrane of a bacteria or an archaea. It's mostly water, about 80%. It does have dissolved solutes in it. There is no distinction or no boundary between the nucleoid region and the cytoplasm. The nucleoid region is simply where the DNA is floating around in the cytoplasm, and there's no boundary between the two. In a eukaryote, we would have a boundary. We'd have the nuclear membrane being the boundary. Please note that the cytoplasm is not all of the liquid portion of the internal part of a eukaryote. We'll talk more about that later. And that's because the plasm inside the nucleus is not called cytoplasm. It's called nucleoplasm. Inside the cytoplasm, you can find the ribosomes and the inclusions. However, ribosomes and inclusions are so big that they are not dissolved in the cytoplasm. Let me see if I can blow that up a little. So here we can see that dot is a ribosome, artist rendition, and you can actually see dots over here for a ribosome. And that's an actual electron microscopic image. Already talked about the nucleoid region. That's where the DNA is located. Oh, I should mention briefly, the DNA does not all have to be inside the chromosome of a prokaryotic cell. Some of the DNA can be in a plasmid, and we'll talk more about plasmids later, but it's DNA outside of the chromosome, at least of a bacteria or an archaea. Actually, the yeast do have plasmids, but most eukaryotes don't have plasmids, but the yeast do. And the plasmid of the yeast is floating around. Oh, I don't remember. Is that in the nucleoplasm or the cytoplasm? Don't remember. I'll have to look that up. Sorry. You don't need to know that for the exam. All right. We talked about ribosomes. Let's move on. You should know that ribosomes are the site of protein synthesis. You should also know that prokaryotic ribosomes are smaller than eukaryotic ribosomes. Prokaryotic ribosomes are 70S in size. The S is where the ribosome sediments in an ultracentrifuge gradient, and the biochemists and the molecular biologists measure ribosomes that way. And the 70S is smaller, the 80S is larger, and so the eukaryote ribosome will sediment lower down in an ultracentrifuge tube. And all you need to know is it's 70S, but that S actually does stand for the sedimentation of the ribosome in the ultracentrifuge tube. The ribosomes are the site of protein synthesis, and ribosomes are made up of ribosomal protein and ribosomal RNA. Ribosomal RNA is also called rRNA. You do not need to know, I won't ask you the makeup units for the ribosome, but you probably should know that a ribosome is made up of two ribosomal units, okay? And you need both of them to get together to form the ribosome. All right, any question about ribosomes? Inclusions are reserved deposits within the cytoplasm. These deposits are used when resources are low or when the cell wants to use them. Inclusions are where the cell brings something into the cell or makes something, and it keeps on getting more and more of it until there's so much of it in one place that it precipitates out of the cytoplasm. Once it precipitates out of the cytoplasm, we can then see it under the light microscope, and it will precipitate out of the cytoplasm. I might have said that twice, sorry if I did. So an inclusion is something the cell is accumulating, and it gets so much of it that the substance can no longer dissolve in the cytoplasm. And then it precipitates out, and that allows us to see it inside the cell. There are different types of inclusions. You just need to know that there are different types. You don't need to know the names of the different types. Here we're looking at some inclusions. You see these spots in the cells. Those are inclusions, and this bacteria must metabolize sulfur, and so it's accumulating sulfur in the cell to such an extent that the sulfur precipitates out of the cytoplasm. These are magnetosomes, and here metachromatic granules. So the yellow is showing you the cell here, and then there are 3 blue inclusions in that cell. Any question about inclusions? If not, let's talk about endospores. Endospores are a special structure that allows the cell to rest and then survive the harsh environment. An endospore is only made by certain species of bacteria, mainly in the families Bacillus and Clostridium. And the point of this endospore is for the cell to survive in an endospore when the environment becomes detrimental to the periental cell. We have a term for that periental cell, and we call it the vegetative state of the cell. The vegetative state is when the cell is metabolically active, doing its thing. It might be moving if it has locomotion. And it's metabolically active, and it's reproducing. And whenever the environment is favorable, that's what the vegetative cell will do. But once the environment starts becoming unfavorable, these cells will start to make an endospore. And then when the environment becomes harsh, and the parent cell will die because the environment is no longer suitable for the parent cell to live in, the endospore can survive in a resting state. And it's just waiting until the environment changes. And once the environment becomes favorable, the endospore can germinate and then return to being a vegetative cell, being metabolically active and growing. As I stated, endospores are resistant to degrading environments. They're also resistant to desiccation, heat, chemicals, radiation. Endospores can survive conditions where the vegetative cell cannot. Endospores are very difficult to kill. You can kill them, but they're not easily killed. And the cell is just waiting around until the environment becomes favorable. Sporulation is the process of making an endospore, and this parent cell made this endospore here. Germination is when the endospore germinates into a vegetative cell. And if that's the case, there will no longer be an endospore. Any questions about any of that? All right. You should understand that an endospore is a special structure that some cells, mainly Bacillus and Clostridium, do to help the cell survive. It is not reproduction. This cell here makes one endospore, but then the parent cell will die. And so we have one parent cell and it'll make one endospore, and then the parent cell will die, the endospore will survive. So we only have one cell. That's not reproduction. 1 to 1 is not reproduction. Any question about that? So to make an endospore, the cell actually goes through division, but instead of dividing down the middle of the cell, which bacteria normally do, the division happens off towards one of the sides. The DNA will get replicated, and then one copy of the DNA will move into the region where the endospore will form. The plasma membrane will then grow and divide this cell into two. We'll then get a double membrane, meaning two membranes to make the endospore. Peptidoglycan layer will then be put in between the two membranes. The spore coat will form. And now this endospore is highly resistant to most things that kill cells. What am I trying to say? Antibacterial agents. And then the parent cell, when it dies, will release the endospore. Any question about any of that? All right, if there's no questions, I'm going to end here and we will show the video next time. I'll see you at 6:30 for the lab.